RNA page. So let's just sort of focus ourselves on where we are in this unit. So biochemistry. We have talked about 4 of the organic molecules that are really important in living things. We start off by talking about carbohydrates, then lipids, then proteins, and finally nucleic acids. We talked about one type of nucleic acid, DNA, and we briefly described RNA. So RNA is also a nucleic acid, so it's similar to DNA in some ways, but also has some differences. It's still made of repeating nucleotides. That's the monomer for this large polymer molecule. However, RNA only has one strand. DNA was a double-stranded molecule. It still has a sugar, a phosphate, and a base in each nucleotide. The sugar is different. It's ribose versus deoxyribose. And the bases are similar. The 3 of them are the same, cytosine, guanine, adenine, but then there's a substitute. DNA had a T, thymine, which was complementary to adenine. In RNA, there's a difference. There's uracil instead of thymine. So there's actually 3 different types of RNA that are important in living things. They have an abbreviation, they have a letter before them: mRNA, tRNA, and rRNA. And all 3 of these types of RNA are important in producing proteins, in protein synthesis. So that they stand for messenger RNA, transfer RNA, and ribosomal RNA. And these 3 nucleic acids work together to basically execute the DNA's instructions. So we know that our DNA is made of those nucleotides with a series of bases, A's, T's, G's, and C's. Your sequence of bases is different from someone else's. That's what makes you unique from somebody else. Those bases, what they actually are, they are the recipe for how to build the proteins that make up a living thing and when to build them. That's really what our DNA is. We talk about our genes. We're talking about the instructions stored in our DNA about how to build the various proteins, when to build.
What's the one thing that changes us, like from like a chimp? Because you said like we're supposed to do a chimp. Like, are these the same as them too, or like, what's
So if you compare the DNA of 2 different species, you'll find species that are closely related have more similar DNA. Humans and chimps share like 99% of our bases would be the same between a chimp and human. There are differences, obviously. And if you compare 2 humans, that obviously the number of differences are even fewer. We'll talk about a little bit more about that as we get to genetics. So the DNA has the instructions to build these proteins, but these types of RNA are needed to make that actually happen. So let's talk about how this actually takes place, and I'll tell you about the role of each of those types of RNA in protein synthesis. So our genetic code is our sequence of bases in our DNA. We have about 3 billion bases that make up our genetic code. So obviously that's a lot. So you can sort of think of DNA, it has the original copy, basically, of the instructions to build a protein. If you thought of like your DNA as like a cookbook with a recipe for how to make some type of chocolate chip cookie, for example. Okay. That's stored in the nucleus, in the DNA, in our chromosomes, which might represent big cookbook. But to actually take that recipe and make chocolate chip cookies, that recipe needs to be brought somewhere else. So DNA contains the recipe to make a protein. Does anyone remember from 7th grade, what part of the cell makes protein?
Mitochondria.
No. Right?
Rhinoplasm.
Yeah, ribosomes. So ribosomes are found in the cytoplasm, attached to the ER, but they're not in the nucleus, right? That's not where they're mainly located. So somehow this recipe about how to build a protein needs to get from the nucleus to the ribosomes. And the molecule that takes that information is messenger RNA. The DNA sort of keeps the recipe safe inside of the nucleus, but it gets copied to messenger RNA when that protein needs to be made. Sort of like if you had this cookbook of family cookbook in your library, your parents didn't want you to bring it out of the library because it contains all of your family's recipes for hundreds of years. You might open up that recipe book, copy down the recipe for how to make these chocolate chip cookies, and then bring that to the kitchen to actually start making the cookies, while the recipe actually stays safe in the library. So the DNA stays in the nucleus, but when that protein is going to be made, the recipe gets copied to this messenger RNA molecule. That messenger RNA molecule can leave the nucleus. Okay? And it can go out of the nucleus into the cytoplasm, and it attaches to the ribosome. The ribosome is actually made of RNA, ribosomal RNA. Because this ribosome is where the protein is going to actually be assembled. So the recipe is there at the ribosome. The ribosome can start to build the protein, but what are the ingredients going to be? What goes into actually making the protein itself?
20 amino acids.
You know, amino acids, and there's 20 possibilities. So the next molecule, transfer RNA, starts to bring in the correct amino acids according to the recipe that's specified by that messenger RNA. These little molecules here are the transfer RNA. You can see they're carrying an amino acid. And so these amino acids start to get linked together by the ribosome, one after another, forming this long chain of amino acids, which we would call what?
Oh, the polymer chain.
It's a polymer, a poly,
Saccharide.
no, not a for amino acids strung together.
Polyprotein.
You're getting it.
Polyacid.
It's polypeptide.
Polypeptide.
Or a protein, we might call it.
Oh, is that to my
And so they get attached one to the other until the recipe has been followed. All of the proper amino acids have been linked together, and then that polypeptide is finished. It can be released by the ribosome and go off into the cell and do whatever it's supposed to do. So the DNA contains the original instructions. The RNA helps to actually execute those instructions and build the proper protein. In biology, we break the process up into sort of two phases. The first phase is called transcription. This is when the DNA instructions are copied first to the messenger RNA molecule. Griffin?
So you don't know if DNA, if like something's wrong, it will mess up like the whole chain. If RNA is something wrong, will it still have the same effect?
Yeah, because the RNA is made based on the DNA. So if you change one of these DNA bases, the messenger RNA that's made from it will also be changed. So remember, one single long DNA molecule has thousands and thousands of genes on it, but only one is sort of needed at a time. So in order to do that, a special enzyme kind of unzips a section of the DNA, and then an enzyme called RNA polymerase starts to build the messenger RNA molecule based on the DNA code. It's called a template. The DNA acts as a template for the RNA. And so the right base is attached, the messenger RNA is created, and then once it's complete, it can leave the nucleus headed out to the ribosomes. So if, let's say, the DNA bases in this area were G, G, C, A, A, T, A, C, C, G, A, T, the RNA that it forms is complementary to it. So what would go with this G in the RNA?
C.
C?
C. G. T. U.
Why U?
Because you complete me.
Right, there's no T, but there is, the U is the substitute. So the messenger RNA bases match up, and then that messenger RNA can go head out of the nucleus to the cytoplasm for the next phase, which is translation. Translation is taking the actual recipe in the messenger RNA and building the protein. The way that the genetic code works is that every 3 messenger RNA bases is a code for one of the 20 amino acids. They're called codons, every 3 messenger RNA bases. And the transfer RNA reads them, brings in the proper amino acids. There are special codons that tell when the beginning of the protein begins, and a stop codon that says, okay, this protein is finished, it can be released. Sorry. You'll have to be able to use this chart eventually, it's on the Regents exam, to figure out which messenger RNA codons code for which of the 20 amino acids. So I'll just show you how to use this chart. So let's say the codon you're interested in is ACG. You look at the first letter of the codon, A. You find it on the left side. So here's A, so it's going to be in this row. Then you go up to the top for the second letter, C. So it's going to be in this column. So it's going to be in here. And then you can follow here, G. ACG is here. That codon, ACG, is a code for the amino acid THR. What about, AAG? What is that a codon for which amino acid?
LYS.
Yes. Lysine, that is. How about GAA? Yeah, glutamic acid, that is. How about UGA? Stop. Yeah, that's the stop codon. So once the messenger RNA has this UGA codon, once the amino acid gets to that, once the ribosome gets to that, it is a signal that the protein is complete and it can be released. So to summarize what we've talked about, the code for how to build proteins is stored in the DNA. To use that code, it's copied first to messenger RNA, which leaves the nucleus, goes to the ribosome. Transfer RNAs start bringing in the correct amino acids based on those codons. They get linked together one after another until the protein is complete. That's the process of DNA transcription and translation. Okay, Thank you.